Hello students, myself Saurabh Yashaswee, Assistant Professor, Department of Zoology, RD and DJ College, Munger University, Munger. Today we are going to discuss about the Hardy-Weinberg principle and its application. So to start with the Hardy-Weinberg, we must know that G.H. Hardy was a mathematician, and along with Weinberg was a German physician in 1908, gave a principle which describes a mathematical relationship between allele frequencies and the genotypic frequencies. So you must be knowing about alleles and the genotype. So when we talk about allele are considered to be points present at the chromosome in a pair, and genotype is the genetic constituent of an organism. So when we talk about alleles, they are always in pair, and so this principle allows us to predict the population genotypic frequencies and its allelic frequency. So let's have a look what is the statement for the Hardy-Weinberg equilibrium, what it states, and it states that both allele and genotype frequency remains constant in a large randomly mating sexual panmetic population from generation to generation until it is not affected by the process like mutation, selection, genetic drift, and differential migration. So these all forces like mutation, selection, genetic drift, and differential migration, I have told you in New Darwinism that these are the forces, external forces, which in long term are capable of giving rise to a new population. So here in Hardy-Weinberg, the population is large, randomly mating, and the allelic frequency of the population and the genotypic frequency will remain constant till this forces does not affect the population. So let us see that how it can happen. So when we talk about a Mendelian population, then we are talking about the fusion of male and female gamete, and the zygotic frequency expected in next generation from random gametic union. And this zygotic frequency is predicted by the gene frequency in the gene pool of the potential population. So suppose if we have a hypothetical population where capital A is one allele and is denoted by small p, and the second allele is small a and it is denoted by small q, then possible chances combination of A, capital A and small a gamete is shown in the next slide by Punnett square, and here we see that capital A, capital A, capital A, small a, capital A, small a, small a, small a. So here p square, that is the genotypic frequency denoted by capital A, where we see that p square is a fraction of next generation expected to be homozygous. Similarly, here we have pq as well as pq, that is 2pq. It is the fraction expected to be heterozygous or capital A, small a. Similar fashion, q square is the fraction expected to be homozygous, that is small a, small a. So the zygotic combination predicted in randomly mating population may be represented as p square plus 2pq plus q square equals to 1. So when we compress this binomial equation, we have p plus q whole square equals to 1, that is p plus q equals to 1. So you here you have to make an attention that it is not minus due to the faint, the sign is quite faint here, it is plus. P plus q equals to 1. So now this p and q can take any value, can have any value, as long as they sum up to 1. That means p plus q should be equal to 1. Now let us take one example and solve a question so that you can have a better understanding about this. So we talk about here the MN blood type. So this blood types is determined by two alleles M and N on the gene of chromosome 4. So when you look at LM, it produces M blood type, LN it produces N blood type, and the heterozygous Mn blood group type is denoted by capital L M and capital L N. So this is the uh, genotype which denotes the blood group. So next, suppose if we give a question and look at how these frequencies are calculated. So suppose in a population like this of 600 129 individuals, we have blood group, as I said in previous slide, M, N and Mn, and this is the genotype. The middle column shows the genotype, and then we have the number of individuals which shows the concerned blood type. For M, in the table you can see it is 1787. For Mn it is 3039, and for N it is 1303. So this is the number of individuals which shows M, Mn and N blood groups respectively. So now in question what it asks, that estimate the frequencies of LM and LN allele. So we have to estimate the frequency of individual alleles LM and LN. Similar fashion, we have to, if the frequencies of this allele has been calculated, so we have to predict the genotype frequencies. So in the next slide I will show you how to calculate the genotypic frequency of Mn blood type gene, and then we will see whether the calculated data allelic frequency really fits our prediction or not. So we should move to the next slide, and remember the first thing we have to estimate is the frequencies of LM and LN allele. So to solve this problem, here we have a population of 6129. So the total number of alleles in a population will be 12,258, right? So population is 6129 and number of alleles were 2. So total number of alleles will be 12,258, because each individual in sample carry 2 alleles of blood type locus. So how to calculate the frequency of LM? So LM is present in homozygotes as well as in form of heterozygote. So when we look here, the 2 times of LM homozygote plus the number of LM LN heterozygotes divided by the total population will give the frequency of LM. So here you see, if you see in the previous table, then the LM, the M blood group population is 1787. So we multiply it by 2 and then we add the heterozygous population, that is 3039, and then finally within the bracket we divide it by 12,258, and this gives a value of 0.5395. In a similar fashion, if we have to calculate the frequency of LN, then we have LN as homozygotes and LM heterozygote. So 2 frequencies, that is then LN 2 frequencies. So we multiply it by 2 into 1303 plus we add the heterozygous population, that is 3039, and then finally we divide it by 12,258, which is the total population, and we get the value as 0.4605. So this is the frequency of LM and the frequency of LN. So we have calculated it as LM as 0.5395 and the frequency of LN is 0.4605. Now the next question is that we have to calculate. So here we have calculated the individual allele frequency of LM and LN. Now we have to calculate the genotypic frequency. So we will move to the next slide, and then we see here that if we have to calculate the genotype, so what is the genotype? It will be LM, that is, in as we saw in the Punnett square, AA, that is the genotypic frequency. Similarly here it is LM, that is the P square. So 0.5395 whole square, that is 0.2911. So for heterozygous it is 2pq. So 2 into p value is 0.5395 into q value is 0.4605 calculated from the previous slide. So we have 2pq, that is 0.4968. In a similar fashion, ln for genotypic frequency is q square, which is equals to 0.4605 whole square, that is equals to 0.2121. So please mind it that there is no minus sign over here. The font has faded away, and that is, you must remember that it is equals to, not the minus sign. So it is, these are the genotypic frequency. So for LM genotype, frequency is p square, that is 0.2911. LN it is 2pq, that is 0.4968, and for ln that is q square equals to 0.2121. So we have now calculated the allele frequency for LM and LN, then we calculated the genotypic frequency. Then we have to see if this data really fits the original data or not, if the calculated values we have calculated fits the original data or not. So here we have, for LM we have 0.2911, that is calculated from the above table, multiplied by 6129, that is the total population. That comes to 1784.2, which is nearly equal to the population which was given to you on the table, the first slide of the problem. So when we look at LMLN, we have 3044.8, and for LNLN we have 0.2121 calculated from the above table multiplied by the population of 6129, that is 1300. So now we have seen that we have obtained the data. Now we will look that whether this data is correct or not. So remember here that the frequency is 1784, then 3044, and that is 1300. So we will go to our previous slide from where the problem has started, and we will see that whether this population the matches with the original population or not. So here is the original data. So see, it is 1787, right? It is 3039, and it is 1303. So if you remember n, it is 1300, which we calculated in the last slide, and the original data which was given to you was 1303. In a similar fashion, you will see that the data which was given in the problem and the data which we have obtained, there are very less differences. So if you calculate the chi-square, if you go for chi-square test and you want to see that what is the observed and the expected difference, then you can easily find out that the difference is not that much. So this is the last slide, and you can compare easily that the difference what we have obtained from our calculation and what it was given for a population, the observed one, is not much. The difference is not much. So that means the data, the frequencies we have calculated has completely fit with the approximately fit with the original data. So this is all about the Hardy-Weinberg equilibrium, and I have tried to solve one of the problem that is related to MN blood group. In a similar way, you can find the problems related to ABO blood group also. So we will be discussing it later on. For today, we will stop here. So stay at home and stay safe.